expression you might see in a knockout system versus a knockdown system. So even though there are these nice examples in the literature of when you target a gene with active Cas9 and get nearly complete deletion, in fact, the amount of cutting and editing of alleles you get is quite heterogeneous. So, at best, you're going to get genes that were never edited, some heterozygotes, and some complete deletions. This is, in fact, much more complicated in some cancer cell lines where you have more than 2 alleles. You may have a real spectrum of different editing phenotypes, and that accounts for some of the heterogeneity. If you saw in the slide that Luke showed from Bruce Conklin's induced nuclease, there's a lot more heterogeneity in that cell type than there is in the CRISPR-I system, and that's partly because you can't really control what happens when you induce editing with active Cas9. This has important implications in a screen context because you're now looking at a pooled population of cells, and you have to think very carefully about what do I call a hit if my population of 1,000 cells, each of which knocks down one gene, has an array of different alleles being knocked down. By contrast, RNAi or CRISPR-I, while you may have not a complete deletion knockdown, it would at least have a more uniform perturbation. So in principle, you could get an allelic series. So a uniform perturbation and different levels of knockdown. This can actually be useful in some contexts. So this is sort of one of the examples from one of the early screen papers where you can see here's an shRNA knockdown where you have different levels of knockdown or CRISPR knockout. And this looks amazing, but I should say this is a single allele of GFP. It turns out to be a very easy gene to knock out because even frame shifts in this gene cause effective loss of fluorescence. But what you'll see even in this seemingly near perfect scenario is this population of cells that actually weren't edited at all. So this is something that you need to consider in the context of a genome scale screen. You will have some cells that are not edited. Okay. I should also say that both of these systems suffer from off-targets, and I thought I would just briefly kind of compare and contrast some of the useful properties of each of these systems. So CRISPR in general has greatly fewer off-targets. I have an asterisk there because I'm going to show you that at least for active Cas9, there are some very significant off-target effects that should be considered, especially in the context of a screen. Namely that if you have an active nuclease in a cell, it can cause DNA damage, and that DNA damage actually has a measurable growth phenotype and potentially can be a modifier of other phenotypes that you're interested in studying. Having said that, if you look, for example, at what Luke had showed for that single GFP knockdown, it tends to be much more specific than RNAi, certainly. It's possible to completely delete genes. It's much easier to find effective guide RNAs. Not only can you delete genes, but as Luke has elegantly shown, you can also activate and repress gene transcription. RNAi, while it has some limitations, as I pointed out, one of the really nice things is that there's a lot less engineering required. So one of the unfortunate kind of pains in the ass about doing a CRISPR screen is you have to get all of those components into the cell. So you have to get Cas9, you have to express it, you have to get a guide RNA, you have to express it at high enough levels. There's a lot of reasons why you obviously want to do that, but I will say one of the nice things about an shRNA screen is you actually only have to put in that one component, and you still have to worry about expression levels, but it's less engineering. Because they don't actually induce DNA damage, even though they have their own toxicity due to saturation of the microRNA machinery and off-target effects, there's less nuclease toxicity in an shRNA screen. The perturbation can be more uniform. It's possible to target specific transcripts, although of course it does have a significant increase in off-target effects. So these are some pluses and minuses to keep in mind. Some of these apply to the CRISPR-I system, less toxic than the than active Cas9 for sure. But actually, many of these disadvantages are not present in CRISPR-I. So I think increasingly people will be sort of shutting down shRNA screens. Although because of this type of thing, in some cases it's actually really nice to be able to pop in an shRNA and not have to worry about getting Cas9 and a guide in. Okay, so in some of the early work that I was doing together with Martin Kopman and Jonathan Weissman's lab, we actually were at that time building what we had thought was a really nicely improved RNAi system. And so I'm going to just briefly describe this because it actually fed into what we then used to do the CRISPR screens. But essentially, we got around this problem of low on-target efficacy and a huge problem of off-target effects by just making a massively redundant library. So instead of the 2 or 3 shRNAs or siRNAs that people had used in the past. We just put 25 shRNAs, and if you use that many shRNAs, you're bound to have some that will work, and you have enough of them that even if there's an off-target for one of them, you can ignore it because you look at the consensus phenotype of those shRNAs. And this greatly increased our ability to statistically significantly pull out hit RNAs in screens, and of course is possible because of the 2 key technologies that enable really a lot of the stuff that we're doing now, the ability to synthesize these pooled libraries in a complex microarray synthesis platform, and the ability to sequence those libraries at the end. So our platform that we built together with Agilent of course, builds on work from Steve Elledge, Greg Hannon, George Church, and many others who had used microRNA-based synthesis to not just make one shRNA, or the exact same thing can be used for guide RNAs, but to synthesize now tens or hundreds of thousands of different guide RNAs or shRNAs in one shot. And you can clone these, because the oligos are high enough fidelity, directly into a lentiviral vector in about a week, plus or minus. It allows you to do very rapid screening in pooled format. So instead of the arrayed format, which I should have mentioned also comes with an enormous cost and just time to actually screen through those libraries, these pooled screens are quite fast. And importantly for our purposes, the platform is very highly adaptable. So when, after we had spent all this time building what we thought was a great siRNA platform and the CRISPR revolution happened, we could just take a guide RNA and drop it into that exact same lentiviral vector.